In April 2026, a severe respiratory illness began tearing through the passenger list of the MV Hantavirus, a cruise ship navigating the remote waters of the South Atlantic. Medical evacuations brought the sickest patients to South Africa, where laboratory testing identified the pathogen. The victims were dying from Hantavirus. That diagnosis contradicted decades of medical data. Typically, you contract hantavirus by inhaling airborne particles of dried rodent drippings or urine, usually while sweeping out an old barn, a dusty basement, or an abandoned rural structure. Furthermore, the established science dictates that the most common North American strains of the virus do not spread directly between people. You have to inhale it straight from the rodent waste. Yet here, a highly lethal, non-contagious disease, tightly linked to dusty, rural environments, was somehow spreading among passengers in the sterile, contained environment of a luxury ocean liner. To understand why the ship outbreak is so unusual, look at this satellite mosaic mapping vegetation across the United States. In the wild, hantavirus spillovers are usually driven by a chain reaction across the ecosystem, known as a trophic cascade. Heavy El Niño rains trigger a sudden surge in plant life, providing a massive food supply that causes the local rodent population to rapidly multiply. But high rodent numbers don't automatically mean high human risk. Research shows wet climates quickly wash viral particles away. The highest risk of infection exists in arid regions. Notice this 15-day window on the timeline, how long the virus survives in dry conditions, allowing infected feces to accumulate and aerosolize heavily. A different environmental trigger operates in South America. In regions like Brazil, severe biome fragmentation, the cutting down of natural forests for agriculture, forces highly adaptable rodent species out of the wild and directly into human food stores and dwellings. In almost every recorded scenario, human infection requires an overlap of specific climate, complex wildlife cycles, and regional geography. None of those ecological factors exist in the middle of the ocean. The investigation narrowed when public health officials mapped the victims' travel histories. Before boarding the ship on April 1st, the first two patients had traveled through the port city of Ushuaia, Argentina. Metagenomic sequencing eventually confirmed the specific pathogen responsible. It was the South American Andes strain of hantavirus. This specific geographic footprint identified a rare biological outlier. The Andes strain is the only variant of hantavirus documented by science able to transmit directly from person to person through close contact. Here is a structural model of the virus mapping its attack at a 2.3 angstrom resolution. These surface proteins bind to human host cell receptors, initiating infection. Once inside, the virus doesn't destroy cells. Instead, it triggers capillary leak syndrome. The tight walls of blood vessels physically separate, allowing blood plasma to leak into surrounding organs. When that massive volume of plasma floods into the lungs, it results in hantavirus pulmonary syndrome. The patients on the cruise ship succumbed because their lungs filled with their own bodily fluids, severely limiting their ability to absorb oxygen. Treating this syndrome highlights a significant gap in modern medicine. Despite ongoing clinical trials, there are currently 0 FDA-licensed vaccines and no approved direct-acting antiviral treatments for hantavirus. For patients entering severe respiratory distress, the primary intervention is a machine called ECMO, which stands for extracorporeal membrane oxygenation. Because the patient's lungs are too flooded to function, the ECMO machine routes their blood outside the body, mechanically oxygenates it, and pumps it back in. It artificially takes over the work of the heart and lungs, keeping the patient alive long enough for their immune system to fight back. Given the severe lack of medical therapeutics, prevention is the only reliable defense. Public health protocols rely heavily on environmental exclusion, specifically the 100-foot rule. This requires keeping all wood piles, thick brush, and garbage receptacles at least 100 feet away from any human dwelling to eliminate rodent nesting zones. If an indoor space is already infested, you must use the wet method to decontaminate it. This protocol requires opening all doors and windows for heavy ventilation and then thoroughly saturating any droppings, nests, or dead rodents with a 10% bleach solution to chemically deactivate the virus before attempting to remove it. The most dangerous mistake during cleanup is dry sweeping or vacuuming. Doing so instantly launches the infectious, dried viral particles into the air and directly into your respiratory tract. The outbreak on the MV Hantavirus demonstrates exactly how pathogens adapt to changing human behavior. As natural habitats fragment and animal populations intersect with human development, localized rural viruses will find their way into modern, hyper-connected travel networks, bringing remote ecological threats directly into global circulation.